Hey, I have a question. By chance, if you are accessing this with the live Zoom recording, can you just tell me that you can hear me well? Because we had a uh, problem with the audio. So I set some of the audio settings that I was told to, and let's see. So if who is here? Uh, Ashima, if you can hear me, can you just type in that it is okay, that you can hear me clear and loud? Let's see. Or some of you in the back also can probably access and see that whether I can hear me well or not. Yeah, it is very faint, so that is one of the issues, and I have no idea. Microphone 4. Is anyone seeing it now? Uh, can you hear me? Okay, okay, very faint. Okay, is it better now? Let's see. It's still really low. Have to. Not any better. Uh, Let me just go to the Zoom and take a look at this. Check, microphone check. Check. Check, microphone check. Check, microphone. So. Auto. This is. This is being an issue. Uh, any better now, guys, folks? Little bit better. Okay, but still, yeah. I am not sure that why this is doing this. Let me see if this is a little bit better or not. Yep, I mean, like I just tried the entire. I see that it is capturing very low input. I see that absolutely, and check microphone check. Yeah, see that it is very low, and do not want to touch that, but I see that what is happening here. Is it better now? No, it's the same thing. Check, microphone check. Oh God. This is Yep, this must be a technical problem still. Let's see. Microphone. These are managed by administrators, so I really cannot do anything with this. What? Made it worse. Okay. So that's the only thing I changed is the USB 4. Earlier it was same as system. This is better or no? Is it better now? No. I mean, like this is a problem. So I think that we will lose class time. And let me just go over and start this, and I will just make sure that I get someone in here for the review. Otherwise it's really, I mean, like I will just process this and still upload that with the Camtasia, and these are some of the things that I have no idea that why this is so I will just double check this for sure. Okay, all right. So let's see. Okay. All right, so let's start. Unfortunately, this is what it is. Okay, so let's see. I've just got distracted with this. All right, so we are starting the last part, which is essentially the cytoskeleton microtubules. That's the last part, and these are from some generalized idea that how their diameter. And unlike the others, the intermediate filaments and the actins, they originate from a microtubule organizing center, which is also known as a centrosome. This is very specific. You have not seen these actins or intermediate filaments do that. For actin, it helps if there is a nucleation, but that nucleation site could be anywhere. But instead of the microtubule organization center or the mTOP, it is a very specific part of the cell. Sometimes it could be at the base of some of the microvilli and the structures that we are going to see. Electron micrograph, and these are essentially some of the roles: beating of cilia, flagella, nerve cell, red blood cell, flagellar structure, alignment, separation of chromosome. We are going to see that all the organization. These are all good. All right, very important slide. You are going to expect a couple of questions from here. So the first thing what you have, that it is made up of mainly 3 different types of tubulin molecule. The third one not that important. So they are made up of this alpha tubulin and the beta tubulin. And then there is also a gamma tubulin that is only associated with the nucleation when this structure starts forming the chain, or back-to-back end-to-end alpha tubulin, beta tubulin strand or filament starts forming. They start with a gamma tubulin and then it gets out. So there is no point in here. Now, if you just look at the structure, this is very different from the intertwined actin filaments that you have from the plus end to the minus end. They still have a plus end and the minus end. And this is the most important thing. Remember that the beta tubule always at the plus end and the is towards the minus end. So it is beta, alpha, beta, alpha, beta, alpha. So beta is the plus end. That is most important part. Third is that internet state really wired internet and still causing some of these problems. Anyway, so if you look at these tubulin molecules, the alpha tubulin, which is shown to have a GTP, This GTP is not hydrolyzable. This is bound as a GTP. It's never hydrolyzed to the GDP, and it has a structural role in the tubulin in the microfilament formation. Whereas the beta tubulin, that what you are seeing, exchangeable GTP binding site. So when the beta tubulin is added at the beta tubulin end, it's added as a GTP. Remember that very well. GTP, just like ATP. You have the actin, it is added as actin ATP. As the same way as the actin, they also treadmill to the minus end. So both of these molecules will be added here, and when the new molecules are added, are going away, and some of the molecules are going to be shed from this end. So at that, as it's treadmilling towards the minus end, this GTP is hydrolyzed to GDP, diphosphate from triphosphate, and then it can shed from the minus end. And this now, a single filament is known as a protofilament, and then see that a single filament, it's going to be rounding up multiple protofilament, and it's going to make a hollow structure. So it is unlike the two strands of actin that intertwine each other for the filamentous actin. It's not like that. It's a hollow structure, much more broader in diameter. So these are some of the most important parts. So definitely remember that the end, which is the minus end, which is the alpha tubulin, beta tubulin end, which GTP is exchangeable in the beta tubulin, this is not, which does have a role. So these both alpha and beta are recruited at the plus end as a dimer. So when you added one alpha tubulin GTP and beta tubulin GTP triphosphate, and then as it treadmills towards the back, this GTP is hydrolyzed to GTP as it's shown here. All right, so how many of them are in there? So if you just look at the cross, if you count these, there are 13 protofilaments, and this structure is called the singlet. There are some doublets where 3 of them are essentially overlapping. So it is 13. You have 13 here, 3 are being shared with, you have a 10. And then here you have a 13 plus 10 plus 10. So these are the 3 different ways that you are going to find the structures. Here what you are seeing, that this is essentially a singlet structure. So 13 of them in those. Then you can have another one, depending upon where they are. Now, these 11, 15, they are occasionally as this. So one of the most important you take from here, that the beta flagella, and essentially you have this microtubule organization center, you always find there are 3 tubules. So know that where you have, if it is just a microtubule that is used for molecules with the motor proteins on them, they are single. Cilia flagella, you are going to singlet. Triplet are the basal bodies, centriole, or the MTOC. So know this slide very well. This is one of the things that people actually miss. So definitely understand that where you find singlet, where you find doublet, and the triplets. All right, so these are some of the pictures of the mitotic tubule, how the microtubule is expanding around with that. And very self-explanatory, will not spend much time. Stable, long-lived, remain polymerized long time. Sperm flagella, red bulb, marginal band, platelets, nerve cells. Here are the specific areas that you can, and again, property, any structure used by the cell to nucleate organized microtubule. Centrosome is or the cell. And there could be separate MTOCs associated with the dendrites and axons of the nerve cell. So just to memorize, just to recap some of your basic idea, that the dendrites are receiving end of a neuron, and these are the axons, which is the delivery part. The action potential travels through the axon and goes to the next neuron or neuromuscular junction. So these are the 2 different parts. So dendrites and axon, and they may have their own own mTOQs, which are the secondary mTOQs essentially. And the basal body is base of cilia or the flagella. So now you have just a typical animal cell in the interface, and it does say, for example, you have a flagella. This is just a schematic. Here you have the centrioles made of the here. Remember that each one of them will have those 13 plus 10 plus 10, 3 of those which is called the triplet. Here you have a secondary microtubule organization center, also known as a basal body. So just remember that here it is shown just like the same way in here, and then the plus end is towards the periphery of the cell and the minus end is towards the body. The same way the plus end extends towards the plasma and the minus end is always towards the basal body, but the exceptions we are going to see. Now here is a dividing cell, and in the typical phase where the chromosomes are essentially being pulled by the microtubules, motor proteins, which are traveling over the microtubules. We are going to see that in a separate place. So what happens that prior to the cell division, a single mTOQ or the centrosome divides into two. Each one of them migrate to a new pole, and from there it grows the microtubules towards the center of the cell, and anything that grows on the mTOQs, the outward end is plus end. The minus end is always the microtubule organizing center. And then as they are capture chromosomes, then they are moved by the motor proteins. We are going to learn that in a separate places, and we're going to talk about that. So this is a typical distribution of a cell. And then the daughter cell will get one of the mTOQ during the interphase, just one. Prior to another cell division, it will divide into two and distribute those. Now here is a nerve cell. Know this very well. Here what you are seeing, a very unique thing that can actually happen. See that all the time we said that the plus end is towards the outer part of the cell, but there are some exceptions to that. Here you can see that the middle one is the same way as all other cell, but the one at the bottom, they have the minus end on this end and the plus end. So they have a reverse polarity. So the question is that if you just ask that where you see a polarity of a microtubule, which means that the minus end is towards the plus end is towards the basal body or the mTOQ, your example will be that they are found in the nerve cells, essentially. So these are some exceptions that you can actually see in nerve cells. So very straightforward. So this is the same. We talked about the critical concentration, the same way in actin. So same way the microtubule have a lower critical concentration for addition to the plus end and a higher critical concentration towards the end, and the GDP beta tubulin loses their fee and they can be end. Also the same way that we gelsolin, those kind of proteins, those are severe, which actually rapidly depolymerize the actin. Here also we are going to see that proteins which will rapidly depolymerize the actin, because the regular depolymerization just by the GTP hydrolysis is not fast enough for some of the processes. All right, after that we can have these disassembly and reassembly of some of the microtubules. So what happens if you cool the cell 4 degrees centigrade on the interface cell, the microtubules actually disintegrate, and that is how you can actually erode if you just cool the cell. Then if you just remove you just increase the temperature to 37, and then the microtubules will develop. You can do this with another compound, which is a calcimide. If you're taking that, we're going to use that this week, essentially for some scratch wound analysis. You add the calcimide, which will essentially not allow the cell to make this microtubule, and essentially will affect the cell division, essentially. So again, remember that there are two things. temperature that are detrimental towards the microtubule formation, and lower the temperature or calcimine will disrupt the microtubule. So here are some comparison with these. I'm not going into detail of this, but just remember that the microfilament and microtubule comparison, this will be an important slide, not a direct question, but it will be very helpful to summarize both the things. So here, one thing that is very important here. So just remember that calcimine and the colchicine, these are pretty much the same compound. One is the calcimine is essentially derived naturally from the source, from a plant source and a fungal source, whereas the colchicine is chemically synthesized. So, but both of them are the same. But colchicine, which is chemically prepared, does have a property that's unique. So the first happens, it binds alpha beta tubule and it causes the depolymerization. But we already talked about this, that know this very well. Calcimine causes the depolymerization, whereas the colchicine in high concentration causes the depolymerization, whereas in low concentration it actually stabilizes the microtubule. So very different type of thing. So essentially, if we just like to see a cell, essentially we are going to make the cell do a fluorescence spectroscopy afterward, fixing the cell. Essentially what we do before that, if we just want to arrest the microtubule in a certain stage of the cell division or something, we add a little bit of this colchicine, so the microtubule actually stabilize. That means that they see that what happens that from during the cell division, from your metaphase to anaphase to telophase, this happens within 20 minutes, very fast. And just want to arrest and look at a particular picture, I mean, like before you fix the cell on those, the event may not happen. So if you just add a little bit of these colchicine, it will arrest stabilize the microtubules in that place, and then you can fix the cell and watch them as well. So these are some of the, so just know that high concentration depolymerize, low concentration stabilize. There's another drugs also which stabilizes the microtubule is a Taxol. These are anti-cancer drugs. So essentially the question is that why it will stabilization of a microtubule will essentially arrest cell division, or why it is an anti-cancer drug. So what you're trying to achieve is essentially to arrest the cell division so that cancer cells cannot grow. So remember that when the cell division is happening, to move the chromosomes to each of the pole to the daughter cells, these microtubules need to shrink from this part. They need to shrink so that the motor pull them. If they are essentially stabilized and not shrinking, that will arrest the cell division. So that's the function of the low of colchicine and also for the Taxol. All right, so after that we are looking at some of these proteins, microtubule associated proteins, regulate plus and polymerization, plus and depolymerization, and also we are looking at some of the motor proteins. We're going to go over all of these. So the first thing that what you have, depending upon a structure, suppose you have a ciliary structure, depending upon the property of that, the microtubules can be differently. Say for example, if you're just looking at these two structures, there are some microtubule associated proteins like a MAP2, and this has a side arm about 25 nanometer. So each of these tiny dots that you are seeing is extending this MAP2. So within 25 nanometer distance, another microtubule cannot be there. So they are kind of dispersed in here. On the other hand, see that in this cross section of the microtubule, that what you are seeing, tau is the spacer protein, and it is much shorter. I mean, like this is more than 25 nanometer. I'm sorry, I misspoke there. Not 25 nanometer. So this is 25 nanometer. This will be 50 nanometer. Whereas this is probably half of that. And if you have a spacer which is much smaller, see that the microtubules are compacted here, and they are much more closer in here. So again, you may ask that where these different things happen. So think about that they are in many different places where they sense the fluid. If they actually bend too much and the fluid level is low, if they tend to bend much, you do not want them to be very stiff. So this will make a kind of a really flexible structure. This will make a stiffer structure. So depending upon the microvilli or the cilia that you find, the cells can make these spacer protein choice and associate them with a microtubule to make these. So you may see some of the electron micrograph or cryo-electron micrograph similar to these proteins. And just know that if you have a longer side arm or a spacer protein, tubules will be associated or they will be arranged in a much more spaced manner. That's basically what it is, take home. All right, let's do, I think that we just only have only one question today because we had to cover a little bit of ground today. So let's do this. We just discussed this essentially. Should be more than enough. I mean, what you want to achieve, that's it. All right, stopping it. That's good enough. Let's see. Yeah, I mean, like, that is the choice, but why you guys got confused? This is just like a pure, if you just go back, this is pure memorization, nothing else. So just remember that what you are trying to achieve is to stabilize the microtubules so that you can study them. Because if they're polymerizing, depolymerizing, you're not going to study them. So see that right here, the calcimide depolymerize them, not going to work. High concentration depolymerize, but the low concentration stabilizes. I gave high concentration, so that's not the answer. Binds the tube, Taxol basically, that's the correct answer. So this type of question we can ask essentially for this. So now let's take a look at this one. This is a little bit of twist compared to the other. 2 minutes. All right, all good? Last chance. Stopping it now. All right. Yeah, that is correct. So that's absolutely. So you can see this type of question. So basically what it is saying that your wild type X, the length of wild type X is this, but when you fuse it with the GFP, see the length actually increased. And there's the same thing that you go back 2 slides back in here, see that what happens, that MAP has a longer sidearm, so it looks like this. It is a shorter sidearm, it looks more compact. The same, just a rationale in here. This is essentially a short side chain or spacer protein, more compact. This is a much more wider spacer protein, and that's why they will look like this. So you can expect some of these type of questions in your exam. All right. After that, we are looking at some of the proteins associated with the microtubule associated proteins, MAPs, which stabilizes the microtubules, and a spacer protein. Now, some of the microtubule tips, these are known as a tip protein, and they regulate the plus end, essentially. So what you are looking at this picture, and in this data, you do not have to dive into the data, but still I'm just going to just explain that what you are seeing, that the EB1 is essentially a protein that binds to the terminal protein, essentially bind to the plus end. And it is in green, and the tubulin is in a red structure. So you can see that at the tip of the microtubule, you have the EB1, this bigger version of that. So at the tip of the microtubule, EB1. So what you are seeing, that this picture is known as a chymograph. It's in your textbook as well, you can take a look. So this chymograph, what it is showing, that where you are seeing this green structure, that's where the microtubule is active with the help of some of these microtubule associated proteins. Whereas the red one, which is the dotted line, this one is essentially the shortening of the microtubule, and we call the point from where it was grown to kind of shrunk, we call this as a catastrophe, but essentially that's the word that's used. So from the tip, the C means it is a catastrophe, and it is going all the way R. And from there, when it's regrowing, we call this as a rescue. So catastrophe, going shortening in length. Rescue is again actually increasing in length. So when microtubule grows, all these microtubules are being added to the plus end, and this is an oscillation. It is not that it is constantly growing, it is just bumping around with the Brownian motion, and it will just grow, and it is just a dynamic movement. And there are some of the proteins that to help in this process. So these actually essentially promote growth, which is enhanced polarization at the. So we actually looked at that. The profilin is one of the which actually helps to add the actin in the plus end. Here is one of these TIPS protein. And they also prevent the disassembly, suppress the catastrophes of the growing plus end, and connect the plus end microtubule to the other structure and the membrane. And these are some of the proteins, like an EB1 is one of the proteins that are that essentially help with the essentially. So after that, we are looking at the protein that regulate the microtubule disassembly. Just know that these proteins, we have only shown these kinesin 13 and stathmin at the plus end. Both of them work at the minus end as well, which is not shown. So you can just think about just comparison. For actin, we have cofilin and gelsolin, which are the severing proteins that works at the minus end. Here, there is a little bit of difference in here. Minus end is usually disintegrated in the actin, not the plus end. Here, the plus end as well as the minus end both can be depolymerized. And we are going to explain that why. So one of them is a kinesin 13, and this is a directed motor protein. It bends the microtubule, and the alpha beta falls off. And the stathmin is all which enhances the GTP hydrolysis. Remember that beta tubulin had the GTP bound when it is added to the plus end, and then as it moves down towards the
the minus end, essentially, the GTP is hydrolyzed to GDP. But this enhances the GTP hydrolysis, and it is actually broken down much more fast. So the question is that why you need to break down the plus end. Remember that we just can go back a couple of slides here for the cell division. During the cell division, the plus end shrinks as well as the minus end shrinks. So all these proteins act as a plus end, and from the middle portion of the cell, it shrinks towards the minus end, and it can work. So that helps the shortening of the microtubules, and that is needed for the cell division. And these are associated with during the cell division, which are the kinesin 13 and stathmin. So know those very well. So right here, proteins that regulate microtubule disassembly at the plus end. Sorry, I didn't have to go back again. All right, so after that, the last set that what we are seeing, that this, most of it will be like this, is the proteins or the motor proteins. So we talked about the myosin, many different types, myosin 2s, myosin 5s, myosin 6, that goes backward. Here you have just only two classes of protein, and they are very specific except just a one or two, which is the exception. So you have a minus end and you have a plus end. So the kinesins is one class of protein that moves towards the plus end. And the dyneins are the ones which are negative end directed motor. These are way larger than myosin and extremely complex. Kinesin is similar to the myosins, but the dynein is way more different than the kinesin or the myosins, and very different structures. So let's take a look at each one of these. So here you do not have to memorize any of those, but if you just look at each one of them, if you look at the kinesins, pretty much all of them, see that they are all moving towards the plus end, where you have a minus end directed motor. Here you have a kinesin NCD. This one is the exception, like myosin 6. The kinesin nuclear cell division related protein. This is the only one that actually is a kinesin but moves towards the minus end. And see that the rest, the dyneins, all of the dyneins are always towards the minus end. But all kinesins are plus end except the kinesin NCD that we are going to see at certain times. So what we are going to see, that we are going to see that cytosolic kinesins that move things on the microtubules, cytosolic dynein, these move on the microtubules, just the direction varies. And then axonymal dynein, these are associated with the cilia and the flagellary movement, and a little bit more complex. So let's take a look at each one of them. So the cytosolic kinesin, this is a generalized structure, very similar to the myosin. You have a globular head, you have a neck region, a rod-shaped portion, and the tail has a light chain. So one difference, just remember that, and you can just probably note this one down as an important for this slide. For the myosin, the light chains are usually in the neck region, and they are calcium dependent. They are in the neck of the, where you have a club-shaped structure, they are in the neck. Whereas the light chains of the kinesins, they are in the tail end, and they bind to the transport vesicles, essentially. And what we mean by the transport vesicles, that we are going to see. So now you know that when we're going to talk about that we constantly are talking about things going from ER to Golgi, Golgi to plasma membrane, then Golgi to the lysosome, and everywhere. We talked about how vesicles form, how vesicles fuse, but how it will go from point A to point B. These kinesins, the dyneins, essentially attach themselves to these vesicles and pull them on the freeways of the microtubules. And if they need a very specific delivery, sometimes they can hand them over to the myosin-based motor proteins, and they go into the alleyways, which are actin-based, essentially. So look at some of the supplementary videos. Some of them are really funny, but it will help a lot. So here we are looking at some of the cytosolic kinesins. So here, organotransport. Even they can move, or like the chloroplast or the mitochondria, peroxisomes, lysosomes, they can place it from one place to the other. So here with the light chains, they are attached and they just walk essentially on the microtubule, and this is a plus end directed motor. Then other functions, see kinesin 5, this is a bipolar kinesin. What it means that essentially it does have a dual head. That means that both the poles have the heads. So you have a head here, you have a head here. And during the cell division, they attach the heads on two different molecules, and essentially they push each other. And this helps in essentially elongating the cell, because when they are pushing the microtubules, as they are associated, if we just go to the cell division, see that right here, they are in the middle, and they are pushing this microtubule, which in the other hand is going to push this mTOX towards the periphery of the cell, and the cell is stretching in size. We are going to go into that detail later part. And then kinesin 13, we just saw, it is a as well as a minus end, which was not shown earlier. Right here, you have shown the plus end, but remember kinesin 13 works well in the minus end disassembly as well. And this is essentially during the shrinking of the microtubules during the cell division. All right, so here what you are looking at, this is ATP dependent phenomena. Plus end directed, contains heavy light chain, requires ATP hydrolysis, transport cargo to different cellular destination, including the cell periphery. So very straightforward with this. And this essentially look at the cytosolic kinesins movement, and this movement is a little bit different from the cross-bridge cycling with the myosin. So just make sure that you understand that. And there is actually a good YouTube video that I found, which is not there, but I will just link that. That will actually help. Again, we're not going to ask a lot of detail of, but the basic idea is that if you just look at that there is a dual head, and this head is associated with a microtubule, and the head that is the backwards, this is essentially with the bound with an ADP. So now what happens? The leading head essentially binds the ATP. That changes a conformational change of the linker and swing the backward head towards the front. So this is very different. Remember that in a cross-bridge cycle, when the ATP binds to the myosin head, it detaches itself. And when it hydrolyzes to ADP, it goes to another position and binds to another new one. Then the phosphate PI, ADP plus PI, the PI is released and then it flexes. That's the cross-bridge cycle. But here see that the forward foot essentially binds to the ATP, and that causes the swinging of the backward front. And then leading edge now binds to a new microtubule ahead of, and the leading head releases the ATP, and coordinates the trailing head hydrolyzes the ATP, and it becomes undocked. So essentially, that's the entirely different part that how ATP is essentially working with this. So here, when the ADP and PI is released from the linker, it becomes undocked. So whereas the PI is doing the power stroke in the cross-bridge cycle. So I will just post a nice YouTube video. If you look at that one time, you will get that. So essentially, the only take-home message is that what ATP does to that essentially allows the swinging of the backward foot towards the front. On the other hand, the cytosolic dynein, you do not have to memorize the detail of that. Just showing the overall structure is a 1200 kilodalton protein, large protein, and they have these ATPase domain, microtubule binding domain, and a dynactin binding domain. So where it actually essentially binds to the carbon molecules. And these heads essentially bind to the microtubules. There are some really nice videos that you essentially see that how the discovery and the detail of the structural biology went in there, and you can scrub those if you do not want to see the detail of those supplementary videos. And you can see that how essentially the dynein actually moves on the microtubule. So it's structurally determined that. So let's take a look at what we are seeing here. Here we are seeing a dynein molecule that is essentially always there. What is important here that there is some of the associated protein with that. So one of them is P150 GLUT. So what it is doing, just remember that sometimes you see on the freeways these big trailer homes being carried by the trucks, and you have a car in the front and the back and the back essentially telling it's a big load in there, and the P150 glued is essentially doing the same purpose. It's just keeping the dynein in a proper place with the cargo so that it doesn't fall off and movement. This structure is a very complex structure. You can see that there is actin, then there is ARP. This is, remember that we talked about the ARP3, but this is ARP1. Then there is a capping protein, and there another protein which is called dynamidin. And all these make this complex, which is known as a dynactin. So essentially all the people ask is that, do I have to memorize all of these? No, you do not have to memorize all of these. This is the thing that you take home from here. So a cargo is associated with an adductor dynactin molecule that is bound to the dynein, and that structure essentially moves over the molecule. So only the dynactin, these individual molecules are not needed, this dynactin. But remember that there are three of these names of these proteins. They are very close to each other, and each one of them can be used as a distractor. So you have a dynein, then you have a dynamidin, which is this. Then this entire structure is dynactin, and then so these are some of the distractors that just use that. So the question is, this is a side talk, that why the scientists named this as a dynamidin. They actually saw in a cell biology experiment that in a cell, if you just essentially overexpress the dynamidin, it blows the entire part. Like the whole binding of the dynein to the cargo molecules actually falls apart. So essentially they thought this like acts as a dynamite, it just blows the dynein function. That's why they named that as a dynamidin. So that's essentially all of it. So what happens that these chains, that if you look at this, these right here, these chains are associated with this cyclic structure, which is a hexamer essentially. And when you by ATP hydrolysis, what happens that these cyclic structure actually move forward. See that right here, this is number one, it just moves anti-clockwise towards the forward, and that essentially puts tension on the cargo to pull it apart. And then what will happen that the dynein head will actually detach itself from here and then place the foot here, and this will go back again to this essentially. So this is essentially how they move. You can look at some of these videos, and it will actually help in your Canvas, these supplementary videos that will help. So where they are carrying things. So you can see that what are the different movements. So you have the cytoplasmic dynein. Remember that the red ones moves from the plus to minus end. Kinesin in the purple will move from the minus to the plus end. So let's take a look at this right here, endoplasmic reticular stack. So this kinesin is moving this towards the cell periphery, whereas this is an endoplasmic reticulum Golgi intermediate complex. They're moving this stack towards the center of the cell. These are moved towards the center of the cell, pituitary vesicles going that way, and in here, this is actually the kinesin which is moving the mitochondria towards the periphery, the same way that these things. So essentially what you're seeing here, that they move all these different vesicles and structures in the cell and rearrange themselves as the cell is moving. But along with that, what is interesting is that see that this picture and this picture. In here, you are seeing a kinesin molecule in purple going towards the plus end, and a dynein molecule in the red is actually hitchhiking the cargo to go to the plus end. And there is a reason for that. On here, you are seeing a dynein molecule is moved towards the minus end, and a kinesin molecule is hitchhiking on the cargo to go towards the minus end. The reason for this is very obvious. Remember that all the dyneins actually move towards the minus end. So when they're moving towards the minus end, after some time, all the dynein will end up in the center of the cell. Kinesins, they move towards the periphery of the cell. They are moving stuff, and they will end towards the plus end of the cell. If the kinesin reach the plus end of the cell, they are plus end directed motor. They cannot go back to the minus end. So how will they go back to the minus end? So the only way they can go back is hitchhiking these vesicles to go back. The dynein the other way. once they go to the minus end, they cannot go to the plus end to bring another, so they will hitchhike to the plus end. And we are going to see that in a little bit of more detail a little bit later. The unique thing that we were talking about, that how the freeways cargoes are essentially handed over to a small ways or alleyways cargo essentially. So the kinesins on the microtubule is moving this vesicle, and this is extremely fast. Think about that you're driving in your freeway at this much faster. Once they need to go to a perfect destination, at that time they will hand over that vesicle to a myosin, which will move over the microfilament, which is a little bit slower, but it will reach to a precise destination. So essentially, this is these actin microfilaments are you can see that these are the city street ways, and these are essentially the freeways. And it is amazing that how actually essentially work with this process. All right, so after that, what we are looking at, what we call as dynein. These are associated with the cilia and the flagella, and for a human being, the only flagella that you have is the sperm cells. That's the only example of that. These are seen in some of the single cell eukaryotes, like the protist. You can see those. And the cilia, they can be many per cell. They help in locomotion. Single cell eukaryotes like Promotium. Whereas the moving particles or fluid over the surface of the tracheal cell, and we're going to see that a lot of these things essentially are present. They are present in the kidney and all the other places as well. So how they essentially move, let's take a look at these. So the structure is pretty complex. So if you just take a cross-section of axoneme, which is an axonemal cilia or flagella, you see that there is a 9+2 arrangement. What we mean by this, in the center, you do have 2 singlets. Each one of them has 13 of them. And then towards the outer periphery, you have 9 doublets, and each one of them is 13 plus 10, 13 plus 10, 30. So remember that you connect these 2 dots together. That is very important, that where you have seen this much earlier. Right here, see that this is a single singlet. This is in the center, 2 of them with 13 microtubules each, 13 protofilaments each, not microtubules. And then in the cilia flagella, you have the doublet. And at the base of each, where these doublet cilia flagella is essentially growing from, they will have a triplet that we are going to see in the last slide. So just make sure that you find out that where they are, where is what. So right here, all right. So now what you are seeing, that each of these doublets are connected by what we call nexin. We are going to see that right here, nexin. And from each one of them, these green-headed spoke-like structures that is essentially extending, those are the dynein molecules, and we call the outer ones as the outer dynein arms and the inner dynein arms. So what's going to happen, that these are going to essentially bound to the next microtubule and try to go towards the minus end, because they are minus end directed motor. And that will essentially bend the flagella or the cilia, and that motion goes clockwise, essentially goes. And that will essentially cause the beating of these flagella, because the movement starts from here and it just goes all the way down. And let's take a look at how this is essentially happening. Just a structure of the axonemal dynein. These are the ends, and this is essentially the base. Very large protein complex. So what is happening right here, if you look at this, this is the minus end tubule, and this is essentially the A tubule. So see that this is essentially the base. If that, this is the base, and this base is attached to the A tubule. Ends essentially bind to the B tubule, and it wants to go towards the minus end. And as the base is associated with the A tubule and they are fixed, the B tubule is going to bend. And this is essentially the structure. So let's take a look at that, what is happening. So see that right here, you have the A tubule and essentially. So this A tubule is going to bind to the B tubule. See that this is the base, and this A tubule heads, the green ones are going to B tubule, which is next doublet, and then they are towards the minus end, and that's essentially going to contract that portion of the flagella. So here what we are looking at. So here we are looking at the A and the B tubules. There is a technical issue with the A tubules and the B tubules. If you carefully look at that, it shows the B tubule right here and the A tubule, but so even sometimes textbooks they forget. So here you can see right here the structure if you look at that. So A tubule you have 13 and the B tubule you have 10. And then here if you are looking at this structure right here, if you look at that, so right here it shows the A tubule with 10 and the B tubule with 13. So it is not exactly correct. The top one should have been it should be the opposite way, but it's fine. It's no worries. It's a double. So but if you just like nitpicking anything, then this structure is different from that what it is. So here what you are seeing that you have a doublet here and a doublet here. What we are essentially seeing, just take one of this doublet and one of this doublet. This is a cross section. Think about that. Here you are looking into the screen essentially. That is your cross section. The picture that we are going to see, if you go in the side and look from the side, the length of it essentially, that's what you are going to see. So here you are seeing the length. Earlier you were seeing from the top just only this cross section. Now you are seeing the length of the cilia or the flagella, and here you have the head. And see that what happens, this is your minus end of the tubule, detach themselves and move towards the minus end, detach themselves, move towards the minus end. That's what you're seeing. But what happens, all these A and the B tubules, they are attached to each other, this Nexin crosslinker. So they cannot go anywhere past each other, and the only thing that they can do is, so essentially it is trying to move towards the minus end. So that this is actually very important. So a structural study was done where the scientists actually digested these Nexin by a specific protease that essentially digests only the Nexin. This is what happens. You can see that this is where the doublets actually slide past each other, and there will be no movement. So what you are going to see in those conditions, that suppose this is the movement you think, you are going to see that this is going to be because all these doublets are going back and forth. They are not converting it into a motion essentially. So you will see that dynein is actually taking up ATP. It is working, but essentially this cell is not going anywhere because there is no net motion of the. Essentially, the Nexin, the cross-linkers are absolutely required for bending of the microtubules. You're going to see essentially a question in your practice quiz and quiz regarding this as well. So know that very well. Right. So here, this is, bless you. So this is one of the most important slide, and why I have put all of these in like this color. Some people get frustrated with my highlighting skills, but each one of them, one of your exam questions, and a lot of people actually miss this slide. So just make sure that you completely understand that, and just make sure you line diagram and do this. So what we mean by this and what is happening here, this is known as an intraflagellar transport. This is not essentially that you are looking at a cell. So let's take a look at this. Earlier we looked at this structure. This is the movement of the kinesin and the dynein inside a cell. Whereas here what we are looking at, think about that this is essentially a cilia or a flagella. These axonal cilia or flagella, which is required for the bending of these. Those are these axonal. But other than these ciliar structure, there are other structures which are essentially many of the sensing molecules. So we are going to see that. And they have these microtubules, and they bring things from the tip of the cilia, sensing molecules, signals, towards the cell, and then send molecules towards the tip so that they can be released outside the essential. So this is known as an intraflagellar transport. Intra means, remember that, not interflagellar. You have just only or one cilia, and this traffic is from the base of that cilia to the tip, and the tip to the bottom. So just only that part, nothing to do with the rest of the part itself. Essentially, what you are seeing that we talked about, here we are looking at the interflagellar transport. The kinesins are moving towards the dynein is hitchhiking. In here, cargo is from the tip to the bottom. Dynein is moving this, and kinesin is hitchhiking. And right here, this is just the opposite way. So essentially what will happen is that why these two pictures are different. See that what happened, that this started from here, the dynein directed motors, they started from here, and they will go back here, and they will start from here, they will go back all the way in here. So that's essentially what is being shown. So one of the important things that what's in here, that they hitchhike this kinesin too, and in absence to dynein will not be able to reach the tip of the flagella, or the retrograde IFT will cease. So what the type of question that we ask, suppose a person is suffering from a kinesin 2 deficiency, and what will essentially actually happen? Just remember that if you do not have the kinesin, first of all, you cannot move the cargo towards the plus end, and also along with that, this minus end directed dyneins, which are going towards the minus end bringing cargo, they will accumulate at the minus end, and they will not be able to go back towards the plus end. This is basically the absence of kinesin 2. Dynein will not be able to reach the tip of the flagella, and the retrograde IFT will cease. What we mean is from the plus end towards the minus end. So retrograde transport is coming into the cell from the tip, that's backwards, by the dynein. But if kinesin is essentially not working, dynein cannot reach the tip of the flagella or cilia to do this retrograde transport. On the other hand, see that in the absence of functional dynein, the other things happen. So what is going to happen, that in this case, all the kinesins are going to move from the minus end towards the plus end, because there is no dynein moving from plus to minus. So kinesins will accumulate essentially to the tip. There will be no kinesins at the bottom or the base, and anterograde IFT will cease. So essentially it will not be able to carry anything towards the tip or towards the outward of the. So and use this analogy or critical thinking question. So just draw it out, plus minus and anterograde retrograde, you should be absolutely fine. All right, so where you actually see, so we are going to look at these sensory organelle, essentially olfaction, photoreception, and flow. These are all good, but here you are seeing a primary cilia. What you do not have that, you have a A and B tubule, 13 plus 10, but you don't have a ciliary, not have a central doublet. So that is essentially the difference between an axonal cilia or flagella, right here. Remember, this is an axonal. This is a primary cilia or flagella without these. Are present in kidney epithelial cells. They can sense the flow of urine, modulate the outputs, and all those different. There are some disease associated with that. One of them is a ciliopathy. This is a genetic disease, and if what happens that if there is a protein called, there's a PKD1 and PKD2, if there is a mutation in some of the PKD2, what happens that it essentially the anterograde transport of these movements essentially, and that actually this is dominant, and it essentially causes this polycystic kidney disease. So cysts are formed, and 1 in 1,000, this is common essentially. So what you are looking at, that this is a primary cilia, all these different cilia, and see that in the middle right here, remember that this is not axonal, it's according to the, or it just can bend due to the flow of the urine, and as it bends, it essentially generates signal, then can be brought toward or from the base of the cell towards the tip. So these are the back and forth movement. So essentially what happens that in the polycystic kidney disease, where you have this PKD1 mutation, these essentially these structures essentially cannot sense do not have any movement of the primary cilia essentially. So this is one of the important part. So just talked about this and connect this. This is the centriole or the basal body, and see that there are 3 or triplets at the base. And here also another connection. Remember that a lot of people actually forget to do this connection. So that's why I'm just going back and forth many times. Triplets of the basal bodies of the centrioles. You have 3 of them, 13 plus 10 plus 10, and that is what it's showing. It's very subtle, but just make sure that you get that. All right, so ciliopathy is known as a Bardet-Biedl syndrome. What happens, essentially, what happens that there is not really a problem with the kinesins in this process. There is a protein known as a BBsome. This is similar to COP or essentially. And when the BBsome, so the BBsome is essentially the association of with the kinesin 2. So the cells have normal distribution of kinesin. They can associate with the cargo in the absence of the BBsome. See that here you have a problem with the kinesin 2, that it cannot move the cargo essentially. Here kinesin 2 is not a problem, but essentially a cargo is attached with the helminths, and that is not present, and that's why nothing moves towards the cilia. So the sensory receptor will traffic to the cilia by the intraflavicular or IFT. So that's cognitive impairment, retinal degeneration. These are some of these. So last part, that what we are seeing, that this is actually if you very carefully look at that. So just remember that we are looking at these IFT complex A and B, and see that what it is going and what is going. So right here the kinesin is in green and the dynein is in purple. So here what we are looking at, this is the B that is in right here in the blue, and this one right here is the kinesin 2, and it is carrying it towards the tip of the flagella, because this is minus N, this is plus N. It's carrying. See that right here, hitchhiking, dynein is hitchhiking, and this is basically the process. On the other hand, what happens that it is coming down on the other hand, see that these are the cargo, IFT complex right here. This is the cargo that is bringing in. Dynein is now associated with the microtubule, and kinesin is essentially hitchhiking and coming back. So this is your anterograde transport, this is retrograde transport. So anterograde transport is done by the kinesin 2s, whereas the retrograde transport is done by the dynein. And one of the important things that's remember that right here is a cell body. So you have these 2 mother and the daughter centrioles. They are always doublets. So they are doublet means like don't. That means like they are 2 structures. So you have 2. One is mother and the daughter. And now remember this. If you fully look at the arrangement of these spokes, 3 dots subtle. But the basal body has the triplet microtubule, and we saw that as well. You have the triplet microtubule. This is people actually miss this question a lot. that you actually get this very, very well, well as well. So pretty much that's it. So just right here, and I think that's pretty much it. Any questions I can answer? We've never finished early, but still have some of the openings in here. Next, so kind of open still. So if you need, again, remember, just send me an email, and I will fit you for your office hours. I will post all these your iClicker things in one quizzes. You can take over and over. All right.